Hello, everyone, and thanks for joining us for today's webinar, Safety as a Science, Analytics for Trial Safety and Pharmacovigilance. I'm Leslie Bepko, host and moderator for Life Science Connect. With us today, I have Brent Myers, Vice President, Clinical and Transitional Analytics, Philip Ross, Clinical Trial Analytics Champion, and Joy Chen, Senior Clinical Analytics Consultant from
Revity Signals.
Easy for me to say. In the next hour, we're going to cover how to tackle safety and pharmacovigilance, data quality, and collaboration challenges to streamline and better manage studies and clinical programs. We'll find out how CRF safety data review, operational accelerators, and pooled in-house safety data can drive decision-making, how a PV database can provide insights in your clinical programs, and how data and visualization capabilities can extend from CDR to PV. We'll be holding a live Q&A session following this presentation. Feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up on after the event. This presentation will be available on demand afterwards, and you'll automatically receive an email when that's accessible. Now, if you run into any issues, please try refreshing your browser first, and if you need any additional support, submit a question via the Q&A. Now, with that said, I'm going to hand it over to our experts to get us started.
Thanks, Leslie. Good morning, good afternoon, everybody. This is Brent Myers, and I appreciate you taking the time to join us today. I realize that there are quite a bit of virtual opportunities out there these days, and we appreciate you taking the time to join us for this topic. And we look forward to making this interactive through demos, but also a Q&A towards the end. So thank you again for joining. First, really what we're looking to cover today is to discuss through our best practices and how you can get access to your safety data, and really, again, those best practices on how to, really the full spectrum of safety data out there, how you can actually be able to use it and make it actionable to the broad spectrum of roles that are involved across, EDC data all the way through to public data or other aggregated sort of data sources. So really to that end, we've got a great lineup of folks. First, we'll hear from Philip Ross on again, that active safety data review, really think of sort of EDC and other safety case analysis. And really looking at how to best track and, again, get access to those data and make them sort of adaptive and relevant to your questions and your particular role. And then transition to sort of later stage safety data, broader, larger public data sets, and look at signal detection and, again, how that can be adapted to your specific questions. And Srinchuan Joy will talk us through that, and I'll also have a demo for that. And then we'll open up for the Q&A. First, just very briefly,
Revity
is a large global company that I think most prominently right now is in the PCR testing, but also lab supports, other life sciences imaging equipment, and omics, and then
Revity Signals.
We sit on the software and services side of working out of corporate, and again, global company, broadly across pharma, agro, and chemical, and very proud of sort of our achievements really in growing across the industry and helping really a broad spectrum of use cases and research through clinical trials and further. But really what we'll focus on today are, again, those clinical, where we focus in the clinical space. And obviously today, these are the 6 core focus areas that we primarily work in. But really today we'll focus on the data review, medical review, and then pivoting those safety analyses and active trials over to pharmacovigilance and sort of the signal detection space, and really how can you bring together sort of the larger data sets that happen post-market, and really talk through, again, what are our best practices and how to sort of adapt this to your needs and questions. But really to kick us off, first we'll open up with a poll question, and I'll ask Leslie to take it from here.
All right, thanks. So poll question number 1, what is the most pressing safety data review issue that you face. So if you could enter your response there in that pop-up window, your options are slow safety data, distributed data, inflexible tools, collaboration tools, or none of the above. Again, we'll give you a couple of seconds there to answer. And if any of our presenters want to chime in while we're waiting, what do you guys think is going to be the answer to this? What do you think is going to be the highest?
Yeah, thanks, Leslie. Yeah, we normally see, the distributed data and getting access to data, so usually is the sort of the primary sort of focus there. And really just, it speaks to sort of the growing number of sort of safety data systems out there to answer the sort of growing number of questions across trials. I don't know, Philip, if you had any other sort of take.
Sure, that aggregation of data from many places is a key element that because your choices of how to aggregate that data really helps to drive your ability to flexibly query across that data and look for signals or attributes that you otherwise might be hidden in the relatively large trial data that you may have collected. So,
all right, you guys ready to see the results?
Absolutely.
All right, here we go. Oh, there you go, distributed data. All right.
I think this is a good, it falls in line with sort of our experiences. And we're looking forward to sort of hearing any sort of additional comments from the audience too when we get to the Q&A.
Absolutely. All right.
Great. I appreciate that. This is Philip Ross. I will briefly introduce myself for context. I worked for a period of time at Pfizer in the informatics space, where I delivered clinical systems for early clinical research, phase 1 trials, and the like. And so that was from the informatics side, and the system delivery side. When I transitioned to BMS, I had a scientific role as a member of the clinical trial teams, in which I was instead working with IT as a partner to develop requirements for systems for accessing data as quickly as possible and appropriately, merging that data, visualizing that data, asking clinical and answering clinical questions. And so I come with a deep understanding of sponsor experience and pressures in drug development. So I'd like to start by just defining the title terms, safety science versus pharmacovigilance. The terms are loosely the same thing, but in the course of this talk, I'm going to reference safety science as the work by clinical trial sponsors who are working to look at the safety data that's emerging from one or more clinical trials in terms of trying to discover new treatments. Often this takes the appearance of, say, if you're a clinician, actually thinking of each patient or subject in the trial as a narrative, that you want to be able to describe the characteristics of the patient and of the evolving data that allows you to draw conclusions about what that patient or subject is experiencing. In contrast, pharmacovigilance is, in the way I'm choosing to define it here, it's looking at spontaneously reported adverse events that can occur either during clinical trials or during usage in heterogeneous populations over a longer period of time post-approval. This data is much larger, and Joy will follow me with a demo of some of our experience looking at this. The data types are quite different for these two. So for safety science, for clinical trial databases, often we're looking at CRF data, case report form data, that has been collected in electronic databases. We may be looking at other types of safety data that may have also been collected, like potentially diaries or ePro data or whatever. And so a key part of our expertise and the expertise that a sponsor or CRO needs to have is aggregating all of that data for review. The safety data in RAVE or your other source CRF data database is often the most rapidly available, but it may not have been fully cleaned or queried. So you have to take into consideration that it may not be fully reliable. This is not reporting at the end of the trial. This is looking at it as early as possible. In contrast, the spontaneously reported adverse events are during a clinical trial stored in are stored in a database like Argus or RSG, either during a clinical trial or during real-world medical use of a sponsor's drugs, for example. And the WHO global database of spontaneous adverse events is available as Vigibase, which has been collected at the Uppsala Monitoring Center and is one of the largest collections across the industry of adverse events. And Leslie, we're at poll question number 2.
All right, so poll question number 2, which tools do you use to analyze clinical safety data? You can select any that apply. So that pop-up should be up there right now. Your choices are Microsoft Excel, eClinical Solutions, the Illuminate, Business Intelligence Tool, Analysis Tool, Exploring Argus or RSG data, or Pharmacovigilance Analysis Tool using external data. So give you a couple seconds here to answer that. Again, to my experts, what are you expecting to see here?
Yeah, I think obviously there's probably quite a bit more, there's even more options than sort of what we have listed here. So if there's something we don't have listed, I'd like to hear about it, in the Q&A. But I think generally sort of as we start to see the results come in, tipping towards the first response there, I think that's where we'll see sort of that pan out. But I do think it will be interesting to see if there's a deviation from the norm this time around. But right now it seems to be on trend, sort of Microsoft Excel, SAS, R, MATLAB. I'd be kind of curious to again here in the Q&A if there's any additional sort of strengths or weaknesses or pain points that come with each of these sort of options.
Yep, and we do have one comment from Sam. We hear you there. Select the one that you use the most. Sam commented that you can't select multiple. And, we do realize that you may be utilizing multiple. You can select the one that you use the most. But then in the chat there, in the Q&A, why don't you guys let us know if you're using something different, or if you're using multiple, what you prefer over another. Fritz is asking about TrueLab. Like we mentioned, there are others. Just looking to see, what you use the most. So we ready for the results? I'll
Yes,
take that as
please.
a yes. All right. Here we go. So Microsoft Excel. And that was actually what I was going to guess too. The Microsoft Excel SASR or MATLAB. So I got one right.
Yep. No surprises there.
Yes, thank you. And of course, that includes SAS, R, and MATLAB. SAS and R are workhorses of the pharmaceutical industry for analyzing data using scripted analysis. And actually, part of the advantage of the alternative visualization tools, such as Spotfire, which we'll be demoing today, are that they don't require the upfront programming that's required for the more rigorous analysis using a scripted language. and are much more, say, intuitive for use by, for example, a clinician or any other person on the clinical team to explore the data and perhaps look for signals or trends that otherwise might not be visible when the statistical team generates those visualizations that you predicted would be your most important visualizations, but instead that there might be some unexpected results. So with that in mind, allow me to do a quick demo of one example of clinical safety exploration. And so for this, I'm going to use the clinical data review package module of Spotfire analyses. I'm not going to talk a lot about the aggregation of the CRF adverse event data into from the electronic database into the Spotfire tool so that these analyses are available. But the point is to take a look at the use of these scientific visualizations. They've already been created, so you can you have them available for your use, and you can modify or extend them. You can filter the subjects in unexpected ways. Unlike perhaps prior SAS or R coded programming, you don't have to try to predict everything that you're going to be looking for. You can explore for things that, as I said, may or may not be expected. So as a brief demo of this, give me just a moment while I bring this up. And so this is the cover page of this Spotfire build. And it is essentially a table of contents. It helps you to look for some of the tabs of interest, some of the pre-built visualizations that are already present in the build and that allow you to ask some questions. And so, for example, let's say I want to look at demographics. And so I go to the demographics page, and I can immediately tell some characteristics of the study. I probably already know how I designed the study, but this actually confirms that I have 28 demographic records, 12 of which are placebo, 11 of which are low-dose study medication, and 5 of which are high-dose study medication. I see the ethnic distribution or racial distribution that has been self-described by the and collected on the demographics page. I can see gender as self-reported on the CRF page. And so I have a sense of the demographic makeup of the subjects in the trial. And let's say, for example, that I am interested in looking at drug-induced liver injury. And so these are all the subjects in the study. And I, for example, on the left axis, we have bilirubin, twofold elevation of bilirubin above the upper limit normal is the concern threshold in this plot. And on the x-axis, I have alanine transaminase, and the threefold elevation above upper limit normal is the level of concern. So these two data points here are potentially of interest. And if I select those subjects, it turns out that that actually is representing a single subject. And that suggests to me that there's one subject that might have elevated, impaired liver function or elevated liver enzymes at the same, that are a potential source of concern. One of the immediate things you want to look at is the time course of those. So we actually have them on the same page here. Having selected this, I see that the time course of bilirubin begins in the normal range, actually slightly elevated, but and then after treatment, so this is at week 2, I see that the bilirubin is much more highly elevated. The liver enzymes, alkaline phosphatase and aspartate transaminase and ALT are all showing similar excursions or elevations in level on a similar time frame. So the time course of this, the only part of this is a little surprising is that the alkaline phosphatase starts elevated prior to treatment. So this is not definitive to say that this is treatment related. And it's not completely definitive to say that function and liver enzyme are entirely the result of potential drug treatment. So let's take a look at the patient profile for this subject. So what I'm looking at here is the dose, the adverse events, and the labs for the subject that had the elevation in liver enzymes and the decrease in liver function. And what I see is that, in fact, there was an adverse event for hyperbilirubinemia, and that these events occurred during the period of drug treatment. But I also see a fair number of elevated lab levels at baseline. So this is not conclusive to say that this is drug-related, partly in terms of timing. It looks like that this patient, in fact, received placebo. So we cannot say that this patient has any drug-related liver issues. So this is not, I would say this is very unlikely to be a drug-induced issue. It is more likely to be a procedure-induced issue, or even more likely, maybe the patient has hepatitis, liver metastases, the liver enzymes could be explained by exercise, and the subject is 84 years old. So there's a possibility of a concurrent medical condition that could be explaining this. So we have an initial glimpse of that information. The second thing I can do is I can go to the adverse events page, and I can show the data for all subjects. And in this page, I can see there's a large number of adverse events that are ranked in terms of frequency. And if I zoom up a little bit so that I can actually see the names, for example, I'm going to look at syncope here. Syncope is fainting, and it is unexpected for this drug class. By the way, the data I'm showing is a publicly available database. It is for a drug that is a dermatology drug, so it's not expected to cause fainting. And so let's take a look at these severe cases. I'm just going to select the severe cases, and I can see that there's 3 patients that actually have reported syncope. And so if I go to those syncope patients and I look at the patient profile again to see what additional information I can find about those patients. So I have the 3 patients down here. If I select the first one, I can see that this patient actually did receive treatment drug in the first row here. That this patient had a case of moderate syncope that was possibly related to treatment. This possibly related to treatment is based on the evaluation at the site. And later, so that occurred at 2 weeks, and then at a later date, the subject had a severe instance of syncope that was the site judged as probably related to drug treatment. This initial quick look suggests strongly that this could be that the syncope is caused or related to exposure to the drug. So with no further ado, what I'd like to do is let me close this section and just say that these are just examples of how you can, taking aggregated electronic database data, you can present that data in a visualization tool, in this case, Spotfire. You can automate the refreshing of that data. So this data could be looked at every day upon refresh, and depending on the refresh rate of your database. And the monitoring physician or the clinical team can look at that data and ensure that the study is being conducted as expected, or that whether the study is indicating any emerging safety issues that really require intervention, or that relate to the other studies that might be underway with this compound or that you may need to design. So it is a very flexible way to take a quick look at the data and derive hypotheses or drive your clinical decision-making for upcoming activities. So with that, I'd like to transition to a slightly different example that's less open-ended, and in which case we've actually derived sort of a flow for processing the data for medical review by, for example, a medical monitor. And in this case, we're specifically interested in being able to collaborate with other team members and ask questions about the data. And so we want to identify if there's any safety issues. We want to be able to collaborate with members of the team, and we want to monitor progress in flagged items in that data. So I will again transition to a demo here. So what we're looking at here is the cover page of a build that has this flow chart in the top panel, a workflow established for medical review of safety of a subject particularly. And in the bottom panel has a data dashboard that shows the current status of queried data in the electronic database that underlies this, from which this data is being loaded. And so what I can do is look at adverse events. In this case, I could look at a variety of things, but I'm going to look at adverse events. I'm pretending to be a medical monitor here. And as I click this, I see that there's, again, a ranking by frequency of the adverse events and coloring by severity. And so I could, in fact, take a look at body system or organ class. What I'm looking for is, I have a choice of organ classes I can focus on. And so let me look at cardiac disorders. So I'm concerned about cardiac disorders. Again, this is a dermatology drug. We shouldn't expect cardiac disorders that are related to drug treatment. And so we're clearly concerned if those are emerging. So if I take, for example, the atrial fibrillation and look at the patient profile for that subject, I can see that it's severe, possibly related to treatment. And yes, the patient was initially treated at a lower dose of treatment drug and then escalated to a higher dose of treatment drug, at which point atrial fibrillation and severe dizziness both occurred. So I am really concerned at this point as a medical monitor that both of these conditions are related to treatment, and I want to be very careful to ask questions and understand what's going on with this. So I can take a look at this adverse event. What I've done is I've gone to an AE listing page. This is part of the structured workflow. I can select the adverse event. I can put in a comment that I can target to a potential person that I want to ask. And I say, I have a question about this individual. I'm seeing some atrial fibrillation and dizziness. What are your thoughts about this safety? Or as I put, comment test here. And I submitted it, and you can see it's now a portion. It's been updated. The other thing I can do is, if I've queried this, I can mark it as queried, and it's now indicated as being queried. And if I go back to start, let's say I can go to the dashboard, and I can see that the latest query was very recent, and there are 9 open queries for this query. So this workflow allows you, the medical monitor, or other members of the team, to query a particular data type, to go through a set of pre-structured queries of the database, and then it makes an entry. It reads from a database, from the electronic database, to, a table that monitors what adverse events have been queried and reflects that in this dashboard. So with that, I'm going to stop demoing, and we go to poll question number 3.
All right, thanks. So what capabilities in conjunction with your analytics solution are the most important to you? Answer all that apply. So that should pop up on your screen right now. You should be able to select more than one, so please do. So to my experts, what do you think we're going to end up with here? Modular safety analytics across use cases, collaboration capabilities, safety data, pharmacovigilance? What do you think?
Yeah, thanks, Leslie. Yeah, I think, keeping in line with sort of what we saw in the first 2 polls, I think it'll end up around sort of the data aggregation, in the sense that, it seems to be like what we see out with a lot of our partners is this continued sort of, we'll say, explosion or growth in the number of data sources, again, just because of the need to specialize in what data needs to be collected for, the specific studies or therapeutic areas, or to answer sort of regulatory questions. But, being able to do that and then get all your data back together in a central, aggregated manner, continues to sort of be that primary challenge we see. Sort of seeing the results come in, it looks like we're getting a pretty broad spectrum here.
All right, just a few more seconds here. Advance.
Yeah, and I think it's, I just want to reemphasize that, that all of these are challenges that we face, and that we often, during clinical trial execution, are focusing on safety data aggregation and trying to evaluate what kind of emerging safety events, what that's trying to tell us. And we will transition after this question to a closer look at pharmacovigilance.
Okay, well then let's see what we have here. Oh, that's a lot more equal than, what do you guys think about these numbers?
Yeah, I think, yes, surprising and sort of the I think equality, a spectrum across all 4 answers. But I think that, the the modular sort of safety analysis sort of goes hand in hand sort of with our final option as well. But yeah, I mean, I hope we can get some, we'll see some sort of more color to these sort of answers, you know, in the Q&A portion, or if you've submitted the questions already in the Q&A, look forward to sort of discussing those with you. But, you know, appreciate the responses.
Okay, and just to dovetail onto that, please, if you do have questions, submit them via the Ask Question window at the bottom of your screen, and we'll get to as many of those as we can.
Great. And with no further ado, I'd like to introduce Joy to talk about some of her work with others here on pharmacovigilance.
All right, thanks, Philip. I'll take it from here. And hi, everyone. I'm Joy. And in the following presentation, on what is pharmacovigilance and why do we need pharmacovigilance. So here is the diagram illustrating the safety monitoring throughout the life cycle of a drug. So before a medicine is being authorized for use, evidence of its safety and efficacy is limited to the results from clinical trials with restricted conditions, where patients are selected carefully and follow up very closely under control conditions, which means that at the time of the medication has been authorized, it has been tested in a relatively small size of patient population for a limited duration. However, after the drug has been approved, the drug may be used in millions of patients for a longer time period, and it's highly likely to be also used with other medications. So the condition of use have been changed. Patient compliance is variable, and inclusion-exclusion criteria are not as restricted as is in the controlled trial settings. So certain side effects may emerge in such circumstances that has not been discovered or reported during a clinical trial. So it is essential that the safety of all medications is being monitored throughout their use in healthcare practice. So compare with the drug safety model that Philip mentioned before, the drug safety model is gathering data during clinical trials, but pharmacovigilance model is collect and analyze a larger and more complex data sets. So what are the data sets that we are actually monitor for pharmacovigilance? So if you look at the graph on the right-hand side, here you can see that this big circle represents the drug being used in the population. And this is the adverse drug reactions occurring in the population. And this is the adverse event being captured in clinical trials. So the data set that we are going to use for pharmacovigilance is here, that we're hoping that it can include not only the clinical trial data, but also including data from, for example, spontaneous reports, like voluntary communication from an individual, like healthcare professionals, consumers, or mandatory reporting requirements from manufacturers. Also from active monitoring systems, interventional study, like we mentioned, the clinical trials, non-interventional study, like epidemiology studies, also non-clinical studies, for example, animal toxicology studies, or systematic review or meta-analysis from published literature, and also even media. So we want to include all relevant sources, like all of them should be considered in our pharmacovigilance data sets. So the idea is to capture adverse events from the entire population and include all the indications in the database, so that the database is reflecting the true situation in the real world. So, but we have to remember that the existing of a report indicating there's association, but it's not necessarily means that there's a isolation causation. So this pharmacovigilance is actually a process that can proactively monitor, actively searching for and identify safety signals from this variety data source. And here are some examples of public data source that is being used for pharmacovigilance. There are VG-based, FAIRS data, Vigilance. So VG-based is the unique WHO global data sets, and it is the largest database of its kind in the world with over 25 million reports being collected, submitted since 1968. And FAIRS data, it is the US FDA Adverse Event Reporting System. It contains adverse events, medical errors reports, and product quality complaints records that are being submitted to FDA. And also Vigilance, and it's a system that manages and analyzes suspected adverse reaction of authorized medication or drugs that is still being studied in clinical trial in European area. So in order to get the data for use, you will need to subscribe to the public data source provider and also go through a series of data curation process before the data is being ready for use to generate signal for pharmacovigilance. So when you get the data, what exactly is the thing that we're going to do with this data? So here, let's take FAIRS data, for example. In this page, it displays, this is actually a screenshot from the FAIRS dashboard. And this graph is displaying the number of adverse event reports received by FDA each year. So as of today, there are over 24 million reports received by FDA since 1969. But just in 2021, just for 1 year, there were over 2.3 million new reports being received. Think about this, just within 1 year. Think about the volume. The data has been submitted and received by FDA. So because current authorities, they are encouraging individuals, consumer or healthcare professionals and companies to submit adverse event reports. So the reported case number increased dramatically in the past decades. As you can see in here, that for the past 10 years, it's pretty much every year FDA receives more than 1 million new reports every year to FAIRS data. But in the early years, as you can see, the report case number are not that high. Staff, they can actually review all the reported case and investigate it and to determine if there's a potential causal association exists between the drug and the adverse event. However, with the current volume, as you can see right now, millions of data every year, it's no longer being able to go through all the case one by one. And that's why we need to use like advanced statistical analysis method to generate safety signal to set up the signal detection system as an important tool in modern pharmacovigilance. So what is signal detection? So signal detection, it can actually help pharmacovigilance scientists, drug safety scientists to recognize a pattern out of this large data sets. And a signal can be something that is a new potentially causal association, or it can be a new aspect of a known association. So a new potential causal association, it can be a new unlabeled adverse event for the drug, or the relationship is previously unknown or incompletely documented. And for the new aspect of a known association, it can be some observation of increase in an labeled event, increased number frequency of an labeled event, or increase in the severity. Or they can be a newly identified at-risk population that was not being identified before. Okay. So there are currently 4 major data mining algorithms being implemented to do the signal detection, and there are EBGM, IC, PRR, and ROR. So each of the stats, it's actually a result of a different statistical algorithm, but all of the algorithm were designed to uncover the same type of information. That is to look for a disproportionately high occurrence of report for an event and drug when comparing to the reports for that event in the entire background database. So the difference between these measurements should be small provided the sample size is large. And also, there's no single best method for signal detection. They could be all used as complementary given their strengths and weakness compared to each other. Without further ado, let's take a look at our pharmacovigilance solution and see how does it look like. And let me share. Let's see, how does it look like? What can we see from this? And also, how can we interact with the data? So this is the data mining results using EBGM. This is just part of one page of our pharmacovigilance solution. And this one is showing the result for EBGM, and the result signal score are shown using tree map. But we also provide a detailed table down below here. So in the tree map, as you can see, there are bigger tiles and smaller tiles. The bigger tiles corresponding to a large number of reports that is associated with this specific preferred terms, adverse event preferred terms. And if you see, there are some brighter red color tiles. This means that this preferred term and that drug adverse event is generating a stronger signal. So as you can see, this color is pretty dark. It's alarming red, although that the tile size is really small. And when we hover over here, you can see the tooltip. It's showing that there's only 23 reported case in this database, but it's indicating that the number of occurrence it's happening more frequently than it's supposed to be. So that's why it's alarming signal is being generated here. Right? So on the right-hand side, we provide this filter that you can filter it to the drug of interest or the adverse event of interest. And also down below here, we provide this for you to select desired cutoff threshold for your signal detection. So now let's take a look at some real drug. For example, I'm going to pick a drug named Pandemitrix. Pandemitrix is a flu vaccine, but it was being found that there's a link between Pandemitrix and narcolepsy that was suspected in 2010 in Sweden and Finland, following a number of reports in children and adolescents. So as you can see in here, in the EBGM signal detection, we found out that, yes, this is actually a signal, although there's only 12 reports being collected in this database, but it's generating a really high EBGM score, showing that there's a abnormal, like something happened more frequently than it's supposed to be. Okay, so after finding the signal actually leading to several regulatory authorities to restrict the use of Pandemitrix, and also there are lots of studies being investigated into this association. Although there's no firm established causal association has been determined, but ongoing studies are still looking into this association. All right, and let's look at something that is being found. There's a causal association and it's been taken out from the markets. Let's look at this rosiglitazone, right? Rosiglitazone is used to treat type 2 diabetes. It has been removed from the market in most of the country, and but it's still being available in the United States, although it's been rapidly dropped. The use has been rapidly dropped since the box warning regarding increased ischemic cardiovascular risk were issued in 2007. So if you look at other two algorithms, PRR and ROR, you can see that the other two statistics is also generating very similar signal for this same drug and the adverse event. So what we just did was retrospectively looking at the signal for those two drugs. As we can see, serious study and risk mitigation actions was being done subsequently after they found the signal. Before people like us, like in pharmaceutical company, this tool is designed to help you actively monitor adverse drug reaction events. Like I said, we provide the filters here so that you can filter that to your company drug, and you can filter it to a specific interest of adverse events, and you can also filter to the signal depending on your company decision. So this entire process is actually a hypothesis generating activity that the signal that generated, it does not prove that there's a causation, but it provides a timely detection of signal for marketed product while minimizing the overall monitoring efforts. And also allow the safety scientists to focusing on investigate and assessment for this signal finding. So if the potential causality is determined, the company can also proceed with risk action plans or helping business to decide help business decision, like they can decide if they want to voluntarily recall the drug from the market, reformulate it, and remarket it. So let me stop sharing and go back to our slides. So in summary, actually, just want to summarize what we just saw, and the signal detection is an important tool in modern days, in the modern pharmacovigilance, because it can identify and monitor safety trend in a timely manner and helping prioritize and execute critical pharmacovigilance activities and further facilitate risk evaluation and mitigation strategy development. So that's pretty much what I want to cover for our pharmacovigilance solution. And now I want to hand it over to Brent.
Thank you, Joy. That was a great presentation. Really appreciate it. I think in the interest of time, we will jump straight to the questions. Leslie, could you put up the questions for us?
Absolutely. So we'll go to here just so that you can see the contact info here. While we're asking, we do have a bunch that came in. We're going to get to as many as we can. Narayana asks, thanks for the presentation. One quick question about local lab data. In oncology studies, we use local labs. Units are different for the same analyte, and we cannot restrict that. Do you guys convert to standard result, then visualize it?
Yeah, great question. And yes, whether your labs are local or central, we can pull those data in and visualize them. And then as far as conversion, generally the best practice, we lean, our system will represent whatever data is loaded into it. And generally, as a best practice, we tend to have the conversion done outside of our system, but it does have the capability. We can add in a conversion table to allow it to make those standardized data conversions. So yes.
Okay, good info. Will asks, does the clinical analytics solution need to be compliant with GCP to support trials decision making?
This is
Yeah.
Philip. So if I can take that, Brent?
Yeah.
So I would say my personal opinion is, based on my experience, is that there's a couple of key elements. One is study bias. You want to be careful that you're executing the trial in a way that you avoid any kind of influence on trial execution that would bias the study. Nonetheless, you need to exercise due diligence during trial conduct and make sure that the trial is well monitored. These solutions help with that, with very flexible tools. Because they, while it is possible to actually validate output, it is typically, I think, our recommendation that these are used for having data come up, and so the sponsor is aware of that emerging data and can actually ensure that studies are being conducted correctly in concordance with the protocol, and that any patient issues are being addressed proactively, and that decision making in terms of collecting new information that's emerging and perhaps changing on the fly ongoing or upcoming study designs is, I think, all within the remit of appropriate due diligence. And however, clearly by end of study, you want to have your final analysis be GCP compliant, and and your submission to be GCP compliant.
Okay, that makes a lot of sense. So looking at the time here, we've gone a little bit over, but totally worth it. That was a lot of great information. If you submitted a question and we did not have a chance to get to it, our experts will reach out to you and answer your questions directly. In addition, the emails are on the screen as well as the website. Just a reminder that this webcast will be available on demand. You'll receive an email when that is available automatically. And just thank you to our experts, and thank you everyone who joined us here today. Hope you have a great rest of your day, and hope to see you back here for another webinar soon.